Assalamualaikum dear brothers and sisters. Yes, welcome you all once again to my YouTube channel. Let's join pharmacy. Hi my dear brothers and sisters. Today I am here with the Unit 1 lecture 5. Already in past classes we discussed until lecture 4. Okay, and I hope they are very good for you. Clear? And I understand. I try to understand you the every concept in those 1 to 4 chapter. Okay, now let's discuss about the fifth chapter or the fifth lecture in Unit 1. And my dear brothers and sisters, it is very important. Isolation and preservation of the pure culture. Okay, this question was asked for 10 marks and 5 marks. And I guaranteed that in next semester, the same question will be asked for 10 marks. Okay, I will tell you which question is that, and also with a detailed note explanation for you I will be given in this video. Okay, so watch the video until last. If you are new to this channel, as usual, subscribe, share, like and on the bell notification. Yes, let's start. Isolation and preservation of the pure culture. Here first understand that what is this isolation. My dear brothers and sisters, isolation is nothing but a separation. Okay, isolate or separation, separating the microorganisms, pure microorganisms. Got it? Here the term isolation means preservation of the pure culture, means increase in their lifestyle, life metabolic rate. Okay, their life exhausting, increasing the life exhausting or the life metabolic rate is nothing but preservation. Got it? And yes, the first one is methods of obtaining a pure culture of organism. Got it? Here some of the methods are there, my dear brothers and sisters, for obtain the pure culture of the organism. See here, pure culture contains only one kind of microorganisms. Okay, now only one in the whole agar solution or in the whole any medium. The pure culture means it contains only one type of the microorganism, a special microorganism, a single, a minute or a unit kind of microorganism. It's known as a pure culture. Okay, it involves the separation of the individual microorganism from mixed population. Here this isolation or the pure culture of the organism, what it involves means it involves the separation of the individual microorganism from mixed population. For an example, see here, this is the mixed population, contains a variety of microorganisms. Here in pure culture what we are doing, just we are separating a single microorganisms, okay, from this mixed microorganisms. This is known as the pure culture and it is a characteristic of this pure culture. It also maintains the progenies in artificial mode where no other organism finds to grow. It also helps to maintain their babies, progenies in artificial mode where no other type of microorganism can grow. Okay, where does it maintains? Which pure culture shows it's important? Yes, it is the important characteristic in pure culture. Isolation. Got it? Yes. Now importance of this pure culture. Here importance means here pure culture helps to study colony characteristics, then biochemical characteristics, staining reaction morphology. Okay, it helps to understand the morphology of the bacteria, then characteristics of that bacteria, biochemical characteristics of that certain microorganism, either bacteria or fungi, then staining reactions, morphology. Got it, my dear brothers and my sister? This is the importance of your pure culture. Now yes, let's move towards the 10 marks question. Methods of obtaining pure culture. There are different types of method, but here I will explain in detail the 3 methods of obtaining the pure culture. How can we maintain or how can we get the pure culture media? See here, my dear brothers and sisters, streak plate method, it's one. Pour plate method, second. Spread plate method, fourth one is roll tube method, and the fifth one is micro manipulator method. These are the 5 important methods for obtaining the pure culture, but here in this we will study these 3. Got it? Streak plate method, pour plate method and spread plate method. And you note one point here, liquid medium type of microbial culture generally isolated by serial dilution method. Means liquid medium is bacteria which contains the liquid type of the media. Okay, that can be cultured in the presence of serial dilution method. If here the solid media is present, means the serial dilution method does not exist, or it doesn't act on the solid media. Only it is a special type for this liquid media. Got it? This is the note for you all. Now let's discuss the first type of the isolation method. The first type of the pure culture isolation method, that is a streak plate method. See, my dear brothers and sisters, streak is nothing but a striking. This type of striking is nothing but a streak plate method. Let's study what is that. A small amount of mixed culture is placed on the tip of the inoculation loop or needle, okay, which is streaked across the surface of the agar medium. See here, my dear brothers and sisters, what is this? This is the one agar medium dish, Petri dish plate. Here what we are doing, a small amount of mixed culture is placed on the tip of the inoculation. Now for an example, say this is the one inoculation needle here, the top, see you are looking the loop, it is the inoculation needle. Here what we are doing, just we are collecting some amount of a mixed culture microorganisms. See, this is the tube. Just imagine. Now this is the tube which contains the mixed type of organism. With the inoculation needle or inoculation tube, I am collecting that. Yes, I collected. Now what I am doing on the Petri dish or on the agar plate, just I am scratching. Yes, this is the streaking method. Over. While we are scratching, or while we are doing this type of scratches on the medium, there what happens, the colony will be forms. Okay, that colony will help to study the characteristic of that microorganism. Yes, that's our one type of the pure culture, methods of pure culture. Here they said a small amount of mixed culture is placed on the tip of the inoculation loop or needle, which is streaked across the surface of the agar medium. Then it thins out. This is the thinning out. The bacteria, some individual bacteria cells are separated and well spaced from each other. They get separated and well spaced from each other. By that way we can easily understand the concept. Here it is not the most commonly used method. Got it? This type of the method is not commonly used but preferred. Commonly used not, but we can prefer this method. Got it? Now see, there are two common methods of the streaking. That is, one is continuous streaking method. Continuous streaking, in the name itself indicating we are striking continuously, like in this, continuously from the beginning to the end without any period of gap. Continuous striking. Yes, some different patterns of striking are there. See here, this is the square type of striking, this is zigzag type of striking. Okay, this is the current type of striking. Just for different types I have given here. The continuous striking method is, this method of striking is continuous in the sense that there is no overlapping of a striking zones. Means we are not overlapping on that. Just once we strike, means from the beginning to the end it will stop. Okay, in between it won't stops, which that inoculation needle, and the loop is sterilized only once in the beginning and the end. Got it? This method is usually for the broth culture microorganism. This type of continuous media we are using in the broth culture microorganism. Broth culture means simple, you remember, that is a type of the culture in which the culture is developed in such a manner we are providing different types of nutritions for that culture. It's nothing but broth culture. Culture means it is a shelter for the microorganisms. Okay, for an example, it is a home, the home in which a different varieties of nutrients are present. Okay, this is nothing but broth culture, and this method is specially for the broth culture. Got it? Now the next method is in striking method, that is discontinuous striking method. In this method, striking is discontinuous and the loop is sterilized between the striking. Means here, like this type, it's one strike, discontinued, then again the loop is sterilized, again the one strike. again the one strike, like this way, we are striking where? On the agar medium. Got it, my dear brothers and sisters? This is a discontinuous medium. Okay, between the striking we are sterilizing the loop or sterilizing this needle. You just remember it is the inoculation needle. This pencil is the inoculation needle, which contains on the tip, it contains the microorganism, mixed microorganism. Now usually the principle should be the next to this striking media, but I have given here principle for this strike plate method. The method is based on the principle of striking. Yes, striking. The technique essentially dilutes the number of organism and reduces their density. Basically this technique, what it is doing, it is diluting the number of organism, yes, and reducing their density. By means of this striking, it allows microbiologists to distinguish and isolate individual bacterial colonies. Microbiologists means they study about the microorganism. Okay, it helps to allow to distinguish and separate the individual bacterial colonies. By striking, what happens? Automatically there will be separation of the colony will be occurs, means bacterial colony gets separated and they provide greater space to study. And a colony is visible, a cluster of the bacteria. It is the colony. See here, it is the colony. It contains the bacteria. In the colony, the column shape structure, in the colony it contains the numerous of the bacteria. In the cluster means a group of bacteria are present. We can easily understand the characteristics of that bacteria. Then all the bacteria originate into the same colony, means a different kind of bacteria will originate in the same colony. Now see, A bacteria will be located in the A colony, B bacteria will be located in the B colony, like this way. Okay, individual colonies are pure colonies. Okay, different individual colonies, means see, this is colony A, colony B, colony C, colony D. Now individual colony, these are the pure colonies. Okay, without any mixing or without any malpractice. The pure colony is transferred to another plate to produce a pure culture of only one type of bacteria. That pure colony, okay, we are collecting that pure colony and we are transferring to the another culture, okay, to the another plate to produce a pure culture. Now for an example, see, initially what happened, just remember, just listen. Initially the plate or any tube, that tube contains a different type of organism, different types of microorganism. After striking, what happens? We will collect some individual type of a microorganism. That individual, the separate microorganisms will be transferred to the different type of plate, different types of plate. In the particular plate, the particular organism is present, microorganism is present. Okay, there it can be easily developed, there it can be easily cultured. It's nothing but pure culture, no, only because only one type of bacteria is growing. Yes, this is the method of striking, okay, to obtain the pure culture. Yes, my dears. Now let's discuss about the second method, pour plate method. One of the very important method among all, the very important method is a pour plate method. One more is a spread plate method is there, later we will discuss. Let's study what is this pour, in the name itself indicating pores, porous. In this method successive dilutions of the inoculum are added into sterile petri dish. Okay, here what happens? A continuous means a successive dilutions of inoculum are added where? Into the sterile petri dish. Okay, I will explain you this point later, because you cannot understand this point now. Later you only get and understand that. See here, to this plates means the sterile petri plates, to these plates melted and cooled agar medium is poured, okay, with rotating the plates, which is then allowed to solidify. For an example, I will give you better example, you don't worry. See here, this is the plate into the sterile, it is a sterile petri plates. Melted and cooled agar medium is poured with the rotating plates. Here what happens? It is a plate. To this plate what we are adding? We are adding the cooled agar medium with the rotating plates. it is the rotating, because we are fixing any motor there. Okay, it starts rotating, okay. While it is rotating, no, it contains agar medium. Then what we are adding? We are adding the inoculum means the mixed bacteria we are adding to this rotating plate. How it is rotating? Because of the machine which was kept base to that plate. Why it is very important for rotating means because it can mix it is throughout the petri dish, it can be mixed. So for that reason, okay, the colony forming is very easy here. After incubation, the plates are examined for the presence of the individual colonies growing throughout the medium. Means after incubation the plates means the petri dish plates or the agar medium plates, there we incubate the different types of the mixed organism. Okay, after that what happens? We will easily study the individual colonies growing throughout that media. Which type of colony is growing, what's their characteristic, we can study that. Now the pure colonies are separated, transferred into broth culture, and thus a pure culture is obtained. Yes, we will separate that pure colonies and transfer into the broth culture, and thus pure culture is obtained. Okay, you will get this all points while I will explain the procedure. You don't worry. Yes, let's study about the principle. See, the main principle is to dilute the inoculum in successive tubes containing liquefied agar medium. The main principle is to dilute the inoculum. We have to dilute that inoculum. Inoculum means that mixed microorganism. We have to dilute that in a tubes that containing liquid agar medium. So as to permit throughout distribution of the bacterial cell with the medium. Okay, as to permit throughout distribution, we are distributing this medium or we are distributing that inoculum throughout. How? By means of that diluting. The mixed culture of the bacteria means inoculum is diluted directly in the tubes containing melted agar medium maintained in the liquid state at the temperature of 42 degree to 45 degree Celsius. Okay, see, later I will tell you one example, beautiful example is there. Here what happens? It is the inoculum, means it is the mixed culture of the bacteria. What happens? It is diluted to these tubes. Okay, it is diluted to these tubes. After these tubes will be poured on the different types of the agar medium. Yes, that's only poor type of the pure culture. Okay, later I will tell you while telling the procedure. Just understand this concept. Here what we are doing, we are directly transferring that inoculum or mixed culture of bacteria into melted agar medium, maintaining the liquid state at the temperature of 42 to 45 degree Celsius. We have to maintain that liquid state, okay, at the temperature 45 to 42. Then the bacteria and the melted medium are mixed well. How they mixed well? By means of that rotating machine or rotating panel. We are mixing that. Now let's discuss the next part, that is the contents of each tube are poured into separate petri plates, allowed to solidify and then incubated. Okay, we have to pour that all the tubes, the tubes in the separate petri plates, and we have to allow them to solid. Then at the then afterwards we have to incubate that. Means we have to keep them in the incubation for warmth of that. We have to keep them warm. When the bacterial colonies develop, the isolated colonies develop both within the agar medium and on the plates. Yes, while the bacterial colonies will be developed, at that time what happened? Isolated, the separated colonies developed both, where they develop both within the agar medium and on the medium. In the agar medium also they will develop, and they will also develop in that water medium. Okay, and these isolated colonies can be picked up, they have to remove and streaked into other agar plate to ensure the purity of the stain. If we want to gain excess amount of purity, means the isolated colonies, we have to pick that and we have to strike in the other type of the agar plate or another agar plate. We have to strike that, okay, to ensure the purity of that stain. Now the main important concept, procedure, my dear brothers and sisters. See here the procedure. Prepare liquefied agar tube. Please understand the procedure very clearly because procedure plays some important role here. Procedure only. Prepare liquefied agar tubes. We have to prepare the liquefied agar tubes and allow to cool to 45 degrees Celsius. Also prepare water blanks numbering 1 to 5. Prepare liquefied agar tubes and allow to cool to 45 degree. Also prepare water blanks numbering 1 to 5. See here, what are these? These are the water blanks numbering 1, 2, 3, 4, 5. Okay, and these are the liquefied agar tubes, agar mediums 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Understand? Now, transfer 1 ml of suspension of the mixed culture of organism to water blank tube. Shake well. Here, it is the sample, means a mixed microorganism. It should be transferred to these all separately. Here, from here, we have to take 6 to 7 ml in the sample, 1 ml to this, 1 ml to this, 1 ml to this, 1 ml to this, 1 ml to this. We have to transfer probably to all these 5 tubes which containing water blank. Then mix them clearly, clearly, inversion. We have to invert them, or easily you can mix it like shaking that or rotating that test tube like this way. We can mix that, and this experiment in your practical, I think so. Got it? Now, next what happened? Transfer 1 ml of suspension from tube 1 to the tube 2. Tube 1 to the tube 2 and expect the same process up to 5. Now, what we have to do? This suspension, tube 1 suspension should be transferred to tube 2. Again from tube 2 to tube 3. Tube 3 to we have to pour to the tube 4. Tube 4 to we have to pour to the tube 5. This give better experience to study the culture, to study that organism, because we have to pouring from one test tube to another test tube, my dear brothers and sisters. Now see. Transfer 1 ml of bacterial suspension from each tubes to 1 to 5 sterile petri dish plates using sterile pipettes. We have to use the pipettes, sterile pipettes, to transfer that suspension, bacterial suspension to 1,5 petri plate dish. These are the petri dish, agar petri dish. Then from each test tube, what we have to do? We have to pass that all. From here 1 ml to this, 1 ml to this, 1 ml to this, respective to all the 5 petri dish we have to transfer. Okay, understand, you will confuse one thing here. I have told you you have to pour from 1 to 2, 2 to 3, 3 to 4, 4 to 5. Okay, after that what I have to do? You have to again maintain each test tube as it is. 1 ml, 1 ml, 1 ml, 1 ml, 1 ml. After that what you have to do? You have to pour the respected test tube to this respected petri dish. 1 ml, yes, 2, 3, 1 ml, 4, test tube 4 to the 4th of the petri dish 1 ml, test tube 5 to the 5th of the petri dish to the 1 ml. Then what we have to do? Pour the liquefied agar medium in tube to each petri dish containing 1 ml diluted suspension. Yes. Then rotate the plates gently for the uniform distribution of the bacterial cell and allow the medium to solidify. After pouring all the 1 ml from the test tube to respected petri plates, then each petri plates should be revolved. Each petri plate should be rotated. Okay. Then what we have to do? We have to allow that medium to solidify. After solidifying or after getting solidifying, incubate the plates for 24 to 48 hours. We have to keep that plates in the warmth condition for 24 hours to 48 hours at maintaining the temperature 37 degrees Celsius in the inverted portion. Okay, we have to maintain. It is a very important point. Okay, we have to follow the whole procedure correctly. At that time only we will get a pure culture. Got it, my dear brothers and sisters? Yes. Now see, it is the one type of the culture. It is the one type of the culture, that is a pour type of the method. Now pour plate method it is. After incubating for 24 to 48 hours, okay, all this petri dish, we will get the one. We will at a certain period or at a certain time we will get some of the pure culture organism. Okay, to get some more extra pure type of the organism or microorganism, what we have to do, we have to transfer that petri dish where, or we have to strike on that petri dish. We have to strike on that petri dish. After striking, what happens, there will be formation of colonies on that petri dish. Then that colony should be again collected, they are transferred to the where, again a new type of the agar plate, agar petri dish, agar petri plate, okay, which these colonies. And this is the time consuming process, and not only time consuming, but it is interesting to do you all. We have to maintain your patience. Patience is very important. Now let's study the third one, that is the spread plate method. Yes, in spread plate method, here what happens, microorganisms are spread over the solidified agar medium within a sterile L-shaped glass rod called spreader. Here what we have to do means, just microorganisms we have to spread where, over the agar medium. I have told you, I have shown you the agar medium, agar plates. Okay, there we have to spread that microorganism. Then with a sterile L-shaped glass rod, we have to, with a sterile L-shaped glass rod, we have to spread that. Okay, by using that L-shaped glass rod. Later I will show that L-shape, like this way, man. L-shaped glass rod. We have to spread that microorganism where, on the petri dish, agar medium. Principle: the spread plate technique involves using a sterilized spreader within a smooth surface made up of metal or the glass. It should be that spreader should be made from the glass or the metal. Then a small amount of bacteria applied in a solution over a plate. Then a small amount of bacteria, okay, applied in a solution over a plate. Then we have to apply some of the bacteria where, over on the plate, to study the colony, to study the pure culture. Then the plate needs to be dry. That plate should be dry at the room temperature so that the agar can absorb the bacteria more readily. So for that's why we have to dry that. Okay, now procedure: take 3 nutrient agar plates and label with the name of the organism to be inoculated. We have to take 3 types of the three types of the nutrient agar plates, 3 nutrient agar plates. Okay, label them with their expected names. After labeling, what happens, incubate the plates within a loop full of the given organism. Inculate the plates within a loop full of the given organism. We have to just inoculate that plates within a loop full of the given organism. If you given one organism, that organism should be inoculate on that plates. Then place plate 1 on the turntable, means revolving table. We have to place that plate 1. Then sterilize the spreader by putting it first in ethanol. Okay, we cannot put directly that spreader on that plate. Which plate? The plate which contains the organism. We cannot directly add that spreader. We cannot spread with the help of that spreader. Okay, why? Means it contains different types of microorganism. It gives you errors in your product. Got it? Now sterilize the spreader by putting it first in the ethanol, then on the flame of the Bunsen burner, and cool the rod at 30 seconds. For 30 seconds you have to cool the rod. Now see, let's understand. Remove the lid of the plate and spins the turntable. We have to remove the lid of that plate and we have to spin that table. See here, this is the nutrient agar plate, this is the dilution bacteria suspension, this is the loop full inoculated. What is this? Nutrient agar plate. Just 1 ml or dilute we have to transfer with the help of the loop full inoculation. Yes, we have to add to this. Here down to that we have to keep on any rotating chambers to rotate that nutrient agar plate. This is the concept. Now what happens? Touch the spreader gently on the surface of the agar and move it forth and back to spread bacterial cells on the agar surface when the turntable is spinning. This is the spinning turntable, this is the glass rod. We have to move forth and back. Okay, means forward and backward. Okay, why? Means to spread that all microorganisms, okay, completely in the agar plate. Now next what we have to do, when the table stops spinning, put the lid over the lower half of the petri dish. Okay, once the table stops spins, means what we have to do, we have to cover the lower half of the petri plate. We have to cover half of that petri plate. lower half, either this or upper, means lower half. Then sterilize the spreader again and repeat the same process for the two plates. We have taken the three nutrient plates, okay. Then again you have to sterilize that spreader. The same process should be continued for the next two plates. Then incubate all three plates at 37 degree Celsius for 24 hours. We have to keep that in a warm condition for 37 degree for 24 hours. Okay, after that we have to collect that microorganism, respected microorganisms. Got it? How? After 24 hours. This type of figure you can observe here. See, isolated colonies, separated colonies. Okay, while you will observe in the microscope or any other type of the microscopy, you can understand, you can easily observe this type of the minute isolated colonies on your Petri plates, agar plates. Yes, my dear brothers and sisters, this is the three important type of the methods for the pure culture. Okay, got it? And these are the very important. So for that's why I have told you, or I have explained you only that three. One is a pore type of, and then spreader type of the, and striking type of the methods, pure culture method. Now let's understand methods of preservation of the pure culture. How to preserve that pure culture? Yes, now we already prepared that pure culture. Now how to preserve that? Simple. After the separation of any microorganisms in a pure culture, then it is very important to preserve the microorganisms. Yes. Then some of the methods are there for preserving. One is refrigeration. It is the homemade technique, refrigeration. See, if you bring any type of vegetables or fruits, means yes, absolutely simply you will keep that fruits or any type of vegetables in the refrigerators. Why? Because to extend their lifespan, like extend their life metabolic activities, we have to keep that in refrigerators. In the same way, we are keeping these microorganisms, the pure cultured microorganisms, in the refrigerators. Pure culture can be stored successfully in refrigerators or the cool rooms at 0 to 4 degrees Celsius. 0 to 4 degrees Celsius. This method is applied for short duration, means we can store it for 2 to 3 weeks for bacteria and 3 to 4 months for the fungi. Okay, it cannot extend this period. It is a short duration. Their growth continues slowly, nutrients being utilized and waste products are released in the media, which finally results in death of the microbes after some time. Why it is known as short duration? Means here what happens, their growth will be continuous slowly. Okay, in refrigerator, if you keep that pure culture in the refrigerator, means they show slight growth in that culture and nutrients being utilized. Okay, in that broth culture we are adding nutrients. That nutrients are utilized and generally they will release the waste products. Yes, release in that waste product should be released in the media only, when finally result in death of that microbes after some time. Got it? This is the one type of refrigeration. It is a preservative for pure culture. The second one is a paraffin method. This is the simple and most economical method of maintaining pure culture of the bacteria and fungi. Okay, it is the very common method to preserve the bacteria and fungi. Here what happens, sterile paraffin is poured over the slant of the culture and stored at the room temperature. Some of the Petri plates which contains the culture. Okay, culture means pure culture. To that Petri plates we have to pour the paraffin. Okay, paraffin layer ensures anaerobic condition and prevents dehydration of the media. Mainly this paraffin layer will ensures the anaerobic condition, means it will exhibit the growth of the or the development of the oxygen. Okay, oxygen, it is very dangerous for this microorganism, to the anaerobic microorganism. At that condition, if the oxygen is present, means in the anaerobic microorganism, the anaerobic microorganism will be die. Okay, they require the absence of the oxygen. For that method we are using this paraffin method. Later we will study that in next that is lecture 6 about anaerobic microorganism, how to store, okay, how to culture that anaerobic microorganism. You got this? Paraffin, it will form the layer It will ensure the anaerobic condition and prevents the dehydration. It will prevent the loss of water of the medium, prevents that. Now the next one is a lyophilization. In this method, the culture is rapidly frozen at very low temperature, that is minus 70 degrees Celsius, and then dehydrated by the vacuum. It is again dehydrated by means of the vacuum, and the culture is frozen very low temperature, minus 70 degree. In this condition, the metabolic activities of the microorganism stopped, means no more growth of that microorganism, and it is the one of the long keeping method, means the duration of this method is long. This method can be used to preserve the culture for the years, for 10 years, for 2 years, for 5 years. We can preserve any type of microorganism for the years. That is the lyophilization. Then the next one is a cryopreservation. It is stored in the liquid nitrogen. In your 10+2 or in your HSC, you know this cryopreservation. We are preserving the pollen grains, okay, the pollen grains of the plants in this type of the liquid nitrogen, stored in liquid nitrogen. This method is suitable for the long-term preservation of the microorganism that does not survive freeze drying. Okay, if the microorganism they are not surviving in the freeze drying, means at that time we are using this liquid nitrogen. Here simply the microorganisms of the pure culture are rapidly frozen in liquid nitrogen at minus 196 degrees Celsius in the presence of glycerol and dimethyl sulfoxide. This glycerol and dimethyl sulfoxide, they prevents the formation of ice crystals which may kill frozen cells. Means generally in the refrigerator, see, if you keep the water in that ice cube, means after 2 hours or 1 hour they get converted into crystals. But here in liquid nitrogen, to prevent that formation of the crystal, we are adding glycerol and the dimethyl sulfoxide. Okay, because if crystal form, means they will kill some of the frozen cell. We cannot preserve that. By this method, some cultures retain viability for 10 to 30 years. For 10 to 30 years we can preserve by means of this culture. Now the next one is the storage in gelatin discs. Gelatin materials, means it is the semi-solid type of the material, gelatin, jelly type material, jelly type liquid. In this method, the bacterial strain after growth is suspended in molten nutrient in gelatin. We are suspending the bacterial strain here, okay, where in the molten nutrient gelatin. This method is selective for the various species of Staphylococcus and Enterobacteriate. It is a very special method for this, that is Staphylococcus and Enterobacteriate. Then storage in a sterile oil. See, it is also very important preservative for the organism. Various fungi like Rhizophus, Aspergillus, Fusarium, Alternaria, Penicillium, etc. These can be stored for the longer periods of time where in this sterile oil. Yes, my dear brothers and sisters, this is the video for you all, that pure culture, isolation, how to isolate the pure culture and preservation of your culture. If you like this video, then don't forget to subscribe, and the notes will be available in the description. Thank you. Assalamu alaikum.